And just a small little forward, of course, during this presentation, I'm going to talk about topics that bridge between biology, medicine, and physics. So as I'm a physician, of course, I'm more proficient in medicine, less in physics. But of course, this is the result, this presentation is the result of bridging different knowledge and talking with different experts in the matter. I'll start with a sort of state of the art about research in homeopathy and dividing it in three main topics: fundamental research, physicochemical characteristics of homeopathic preparations. And this is a summary of that. It's a publication in 2019. And out of more than 100 previous publications, the researchers, they reviewed these publications and summarized two groups, 29 experiments that were fulfilling three criteria: blinding, randomization, and inferential statistics, and another group of 10 papers that fulfilled 5 criteria. And what are the results? That examining the homeopathic preparations compared to water or succussed water with conventional devices like spectroscopy or imaging methods or other conventional method, what happened is that in 79% or 80% of the studies, there were reported differences between the homeopathic preparation and the control. That could be water or succussed water. So to say that using conventional devices and trying to figure out if there are any differences in 80% of the experiments, there were differences. So homeopathic preparation behave differently than water or succussed water. So this is very fundamental research that is quite difficult to disprove that. Another field of research is about living models in laboratories, like cells, different line cells, and also vegetal models. And this is a review of several dozens of studies, starting from mother tincture. Of course, with mother tincture, we have material doses of the substance, for example, phytolacca. But then we go on, we go higher in diluting and succussing the preparation. Then we go to the 3x, for example, is a dilution and succussion 3 times 1 to 10. And then we go up again and we get to the 5C, 6C. This is the centesimal potency. That means that you dilute and succuss 6 times in a ratio that is 1 to 100. So the differences reported, for example, with the 5C Lycopodium clavatum in cervical cancer HeLa cells was an increase of apoptosis of the cancer cells treated with Lycopodium clavatum 5C compared to the controls. And so on. We go on with the experiments, and we find experiments using what is called surely highly diluted and potentized remedies, like the 30C Arnica montana at the bottom of the screen, measured with the E. coli and seeing what are some differences. For example, when using arsenicum album 30C the E. coli bacteria more resistance to the arsenic toxicity that was given afterwards the treatment with arsenicum album 30C. And so at this point, we see we don't find molecules of the initial substance of the arsenic that we use at the beginning of the process of diluting and potentizing. And nevertheless, we can see biological effects in cells, and it's very difficult to see sort of placebo effect. And so on, we go up with the 200C and some other cells and some other effects. You can read on the right, an increase of apoptosis or a difference in the gene expression. Of course, always compared with control substances like water or dynamized water. So also in these biological and lab models, we see again, there are many studies that show there are significant differences between the homeopathic preparation and the control, the inert control. This is another researcher, Stefan Baumgartner, working at the University of Bern, and he produced during the last 20 years several experiments using some plant models in the lab. And again, he showed very significant differences in several models, and he replicated many times the experiments. And then we jump to the third main topic about homeopathic research, that are the clinical studies. And we go directly to the meta-analysis, that are the review, systematic reviews and meta-analysis of several studies, previous studies performed with the placebo control randomization. And so we start, and usually from the media, we know that there was a 2005 study published in the Lancet, written by Shang, that demonstrated, they claim they demonstrated that homeopathy was ineffective. And if you go on the right of the screen, actually under the column valuation, and you cross with Shang, and then you find there's a minus sign. But if you go higher, then you see there are only plus signs about the evaluation, meaning that the previous meta-analysis and also another meta-analysis performed in 2014, so starting from 1991 till 2014, 5 meta-analyses have been produced that were all in favor of the efficacy of homeopathy in clinical settings, treating different pathologies. Also, when you find going below the Mathieu in 2014, you find the second Australian report. But this second Australian report actually has been withdrawn after that it has been shown that there were several flaws about this report. And the first Australian report, again, that wasn't published at the beginning, but published after. So it was done in 2012 and then published in 2019, actually was showing that, again, homeopathy was effective, clinically effective. Also, go into different kinds of studies where homeopathy is compared directly, not to placebo, but to conventional treatments. Homeopathy in the yellows, homeopathy appeared to be at least as effective as conventional medical care in the treatment of patients with the precondition studies that were upper respiratory tract complaints and lower respiratory tract and ear complaints, were quite very common complaints. So homeopathy again was as effective as conventional treatments. So now we are confronted with this scenario. Experimental data in the last 30 years are showing positive results, and most of the studies are in favor of homeopathy. Now there's a big question mark. So if we still think that there's nothing in it, there's nothing in the homeopathic preparations, the logical conclusion is that experimental data must be wrong, or either if we usually think there's nothing in it, but we don't think that experimental data, such a huge amount of data, is wrong, then there's something we don't know, there's something we are missing. So let's explore, first of all, the arrow going up. So the hypothesis, all the experimental data are wrong. And this comes out with an article published this year, and very eminent epidemiologists like Ioannidis published with some other fellows this article about homeopathy can offer empirical insights on treatment effects in a null field. So what is actually a null field? Is something where there's nothing inside, and so there's nothing scientifically there to know, and so any observed effects reflect simply the magnitude of the prevailing biases, the noise in the scientific process. So homeopathy, according to these researchers, is defined as a null field. There's nothing inside, and so all the amount of research that has been performed in the last 30 years must be dealing with wrong research. There's a bias, different kind of errors, and so let's explore a bit more what about these kind of biases and mistakes, that allegedly mistakes of homeopathic research, from fundamental to clinical research. For example, this article that also makes reference to the other article on the top left of the screen, says that based on the results of the meta-analysis from 2014 2014 to 2019, analysis about homeopathy. Homeopathy should be classified provisionally in the group of therapies, like conventional therapies, which are effective but need research, because looking at the Cochrane review of systematic reviews of conventional treatments, it's 2007, they revealed that 96% of all systematic reviews call for more methodologically high quality research, and 49 of these publications present results that do not allow any conclusion to be drawn about the benefit of the examined intervention. And when criteria of evidence-based medicine are considered and taken into account, if on all quality levels, according to the Cochrane criteria, are recognizable even in the methodologically high quality studies of homeopathy. So when you compare, when you compare the quality of homeopathic studies with the general quality of other conventional studies, there's quite a good matching of quality. Anyway, Wagner and others, they published 2 years ago a study where they underlined that 46% of registered trials in homeopathy, clinical trials in homeopathy, remained unpublished, and 25% of published homeopathic trials the primary outcome measures had been reached or modified. So there are two problems with quality and bias. When we look at conventional studies, we find that half of all registered clinical trials in conventional medicine report their results within a 12-month period, whereas 62% of all registered homeopathic trials reach publication. And again, inconsistencies in reporting of primary outcome occurs in 43% of conventional medical studies, whilst Gardner and others, they documented that this occurs only in 25% of published homeopathic trials. So the Homeopathic Research Institute, that is a bunch of researchers, very well-trained researchers, they concluded that we now know, thanks to these studies, that homeopathy is outperforming conventional medicine in the respect of bias, with lower levels of reporting bias. But let's go to the article about the null field, homeopathy as a null field. And the authors, they say that homeopathy is the mechanism of action is inconsistent with well-known physical laws. And where do they get this affirmation? They get it, if you look in the reference, they get it from this paper of 2012, published by Grimes, sorry, I don't know how to pronounce it, where he claims that the meta-analysis of published trials has also shown no positive effect of homeopathy, and he mentions only Shang et al. But we've seen before that there are more meta-analysis and showing that homeopathy was effective. Actually, only Shang was saying, showing that it wasn't effective, excluding 93% of the studies. Another claim, this article, is that the few studies that have suggested a weak beneficial effect have been of low quality. This can be disproved by this other article in the left up side of the screen. You can find it. And the table down on the left, it shows that considering all the studies mentioned in the article, you get 1.53% showing effectiveness of homeopathy. When you reduce and reduce and reduce to the highest quality studies, and you get to the B1 that in the studies considered the best quality, the highest quality studies in clinical research of homeopathy, the quality of the proof of effectiveness increases even more. So disproving what is claimed in this paper. Again, homeopathy insists that the biological potency of any homeopathic remedy increases with dilution. That actually is not only dilution, but dilution and succussion. This runs contrary to what is usually observed in nature, where higher doses trigger correspondingly higher reactions. Actually, this is not exact, because if you see, this is very fundamental research and nothing to do with homeopathy. This is research including plant phytochemicals and in cancer research, and they showed a biphasic dose response. So when you increase, and you look at on the right side, and when you increase the concentration, you get higher effect, but at a certain point, you increase more the concentration, the dose of the phytochemical, and the effect goes down. So this is a biphasic dose response, and it doesn't fit with this claim that are not correct. Again, proposed mechanisms are physically impossible, and the author claim that when you go up the 12C dilution, you go higher than the Avogadro's number, actually. But if you look at this research, when where they studied cuprum metallicum and gelsemium as a plant, as a homeopathic preparation, compared to controls, of course, inert controls, they found that active molecules, for example, see on the left side, active molecules of gelsemium that are large molecules, gelsemine or sempervirine, ended up to be found in the preparation up over the 7 decimal dilution, that is corresponding to the 4th centesimal dilution. So what does it mean? That actually, and also they found that even in the highest dilutions, and like 30C to 100C, when nothing is expected to be found in, they researchers found even in this highest dilution the presence of nanoparticles, and so the claim about the Avogadro's limit for homeopathic potentization is invalidated, because homeopathic preparation at 30C is not just water, is a mixture of nanoparticles and water, and this is a very new story, but we'll see afterwards. So the Avogadro's number used to disprove the possibility that highest potentization dilutions of homeopathic remedies can't work. This is a correct theory predicting the number of molecules in a mole of a substance, but actually it's according to experimental evidence is used out of context when used to predict the content of homeopathic preparations. So the theory doesn't fit with the experimental data and vice versa. Again, the previous article, they considered about water memory. So they say, okay, there's nothing in it, but homeopaths say there's water has memory. Actually, we're seeing that in the homeopathic preparations, there's not only water, there are also nanoparticles. They don't allow us to say that nanoparticles create an effect like a molecular interaction with a receptor. There's not that, but we see later what kind of effect they can do. So the author examined the hydrogen bonds of water, thinking that homeopathic preparation contains only water. That's not exact. It's not true. And of course, the hydrogen bonds alone, they last only some femtoseconds. So that's not the way of finding out something in homeopathic preparation. But let's go now in homeopathic theoretical research done by some physicists. And I've taken, with permission from the journal Water, some slides of a presentation given by the physicist Tamar Hinon, that unfortunately died a few years ago. So first of all, when we examine homeopathic preparation, we find that there's supramolecular organization. There's the aggregation of molecules and nanoparticles into clusters. This is something that cannot be explained with customary theories. So there are some problems because we can't use customary theories of liquids to explain why and how these clusters that are experimentally have been seen and verified, how these clusters form, and they are long-lasting. They don't last just femtosecond. They last for years in the homeopathic preparations. So according to the customary theories, foremost model, the interactions between the liquid molecules are electrical interactions, and they do not take into account that actually there's an electromagnetic field that comes from the quantum field theory that all reality is pervaded by different fields, and one of these fields is the quantum electromagnetic field. And so this allows interactions that are not only short range, but also long range interactions, because the electromagnetic field is everywhere, and it can, let's say, make a sort of communication between distant things, like very simple. When you use only the electrostatic model, it's like when you comb your hair and the hair stick high. But when you use the electrodynamic model, it's like if you are on two different boats over the sea, and then you use your radio to communicate. And this communication happens not because of local interaction, but long distance interactions between the vibration of the electromagnetic field. And so what the experimental findings are that homeopathic preparations, they display a series of different electrical and physical chemical characteristics. For example, electrical conductivity is increased by 100 and 1000% in the homeopathic preparations, or also, for example, the pH metric is different, or the conductometric measurement are different. So now there are some slides into the quantum field theory, and they summarize what was the development. Of course, biochemistry and classical chemistry, they just use what are the knowledge, what is the knowledge that comes before considering the quantum here of the 20th century. So what is worth looking that in 1973, two physicists, they spoke about this kind of possibility that deriving from quantum electrodynamic interactions, condensed matter, it means liquids and solids, so also liquids like homeopathic preparation, condensed matter, it can share a sort of super radiant phase transition. That means very shortly, it means that these matter, liquids, for example, they, under specific circumstances, they can show a coherent behavior. This is a key word, a coherent behavior, that cannot be explained only by electrostatic interactions. And going on, in the later years, after 1973, many physicists questioned the physical reality of this possibility of coherence, but it was thanks to some Italian physicists, Preparata and the Giudice Meli, and some other co-workers, that they showed theoretically that the possibility of the formation of coherence domains in different materials, liquids or solids, that oscillate in phase with an electromagnetic field. And this theoretical hypotheses have been confirmed by independent researchers in the following years. And again, the same physicists provide examples of this coherence in a large variety of condensed matter systems, including water, alcohol, and so on. And you can find many references that were included in the Tamar Yinon presentation. If you like, I can send you the whole presentation. Just email me. My email was at the beginning, but I can write it in the chat afterwards. So this is the list of the references. And so let's go back. Very simple, because now we are going too much into the physical world and the physics, and this is not my field. Just considering very few experimental data from fundamental research, we find there are pH changes in the homeopathic preparations different from controls, differences in electrical conductivity, different crystal shapes when examining dried up preparations, and a different relaxation time at the magnetic resonance. So just considering these few data, and there are several other data, but these and other data, experimental data, actually were predicted by the Giudice and Preparata theory. So it's very promising field of research, looking at the physicochemical characteristic of homeopathic preparations, having the theory that you can confront the experimental data and see if the two fit together. Of course, if we go into this field of considering the homeopathic preparations a kind of particular physical entity that is not behaving using the molecular interaction direct, but using this kind of coherent information, then you can ask yourself, but you need also a receptor. So are biological systems receptive? Can they receive this kind of information that is not molecular? And there's a long history of research in quantum biology, and very summarizing the most fundamental evidence that has emerged in the last 10, 20 years is that living organisms display a very interesting set of quantum characteristics that is not contemplated by classical chemistry, but experimentally they are there. Like photosynthesis is so efficient in leaves, in vegetable leaves, plant leaves, because of quantum coherent superposition. Many enzymatic processes that happen even in our organisms, they are so efficient and so quick to happen thanks to quantum tunneling. That is like something crossing a barrier without an increase of energy that can jump over the barrier, but crossing through the barrier. Olfaction, our olfaction has been proven that was using again a quantum tunneling effect, and the ability of birds of orientating and migrate is experimentally confirmed that is due to quantum entanglement and cognition. So our brain activity, Freeman and Vittorio is a Italian physicist that also collaborated with Preparata and Del Giudice, and they've shown experimentally that the brain can do what it does only if we take into account quantum processes that let processes happening in different areas of the brain happening instantaneously, and they write extensively about the Nambu-Gosson bosons that we find later on. So let's go quickly. So as mentioned at the beginning in my introduction, I've written with a colleague of mine 2 articles explaining homeopathy with quantum electrodynamics, and this other that is the quantum nature of biologics. And if the first one is copyright protected, so I'm not able to let you have the full article, and you have to purchase it from the publisher. And the other one is open access, and you can read it fully. These 2 articles, they summarize 40 years of theoretical hypotheses and at least 20 years of research done by many theoretical physicists and many researchers, where we very briefly we say this principle: the principles of quantum field theory, quantum electrodynamics, and gauge frameworks explain multi-level coherence in living organisms in terms of balanced competition between the gauge and Nambu-Gosson bosons as emerging from symmetry breakings. I can understand that these are some weird terms, but I can try to answer with, of course, my knowledge of a physician, not a physicist, but I can try to answer later. Biological intelligence that is used by us as a sort of new proposal instead of force that was quite outdated and thrown out from the scientific community. But actually, biological intelligence can be explained using the theories, the quantum field theories, quantum electrodynamics, is an expression of connectedness, multi-level coherence, and reactive adaptability in living organisms. And the presence of these Nambu-Gosson bosons, condensations, are responsible for order and coherence in living matter, living organisms. So just to sum very briefly, we can say some of what is written in this slide is already well confirmed. Some other parts are still in the hypothesis of the field of the hypothesis, but as a whole, what we are working on as an hypothesis to fully confirm by experimental data is this: on the side of living systems, we can imagine this: events provoke symmetry breaking of the dynamics of the systems. Information of the events is stored in condensation of Nambu-Gosson bosons that are generated after symmetry breaking. And Nambu-Gosson bosons keep the information of the event and image composed of vibrational modes. This is in the living systems. So when we analyze the homeopathic remedies, we can put this hypothesis and partially confirmed by data, vibrational mode of the substance that we can say give the image of the substance are released during the preparation, dilution, succussion, as Nambu-Gosson bosons that are generated as information of the state of the system and stored as vibrational modes of water with nanoparticles and all the stuff that is inside the homeopathic preparation. There's not just water. And a multi-level coherence in living systems allows to model them as quantum systems where entanglement, superposition, and tunneling phenomena take place, as we already have seen from conventional research showing some of these phenomena happening. So I like to dedicate, as Tamar Inon did with a slide, I also like to dedicate this presentation to Professor Giuliano Preparata, that he was a theoretical physicist, a professor at the University of Milan, and unfortunately died in the year 2000. And I like also to remember what once, when questioned about some weird phenomena, and he replied, the true scientist, when observing a real fact, if he doesn't believe it, and I think it's good for a scientist not just believe in it, it means that he hasn't the instrument allowing him to understand how that fact can happen. Instead of ridiculing the absurd fact, the true inquisitive scientist should make any effort to understand what is missing in the conventionally accepted vision of reality, in order to include the absurd phenomena once they are doubtlessly reproduced in the field of scientific rationality. So the main effort is reproduce the phenomena about the homeopathic phenomena in fundamental research, clinical research, and so on, and lab research many, many times, as it has already been done And surely it will be done, and many more publications are going to be published about fundamental research. And when the observed phenomena are surely reproduced, then we must ask ourselves what is missing about knowledge of reality that can explain what we observe. So thank you for your attention. And if there are any questions, I hope I haven't been too complicated. I'm here to reply. If